So I want you to picture a rat in a laboratory cage. But this isn't your standard behavioral setup.
Right, definitely not a normal maze.
No. The floor of this cage is actually an electrified grid. And at the very far end, there's a small metal lever. Now, this animal is starving. I mean, it has been severely deprived of food, so it's biological imperative to seek calories. That should be the absolute overriding priority of its entire nervous system.
That should be the only thing it cares about.
Exactly. Yet when placed in this box, the rat willingly and repeatedly steps right onto that electrified grid. It takes painful shocks to its feet just over and over, all to reach that lever. It completely ignores the food.
Yeah, it won't even look at it.
It will just press that lever for hours, even days, literally to the point of starvation and death.
Yeah.
And what does the lever do? It delivers this microscopic pulse of electricity directly into a specific bundle of fibers in the animal's midbrain.
It is just a devastatingly clear demonstration. I mean, you are looking at a nervous system that has been completely hijacked. The biological machinery that's fundamentally designed to keep the animal alive has just been commandeered to desire its own destruction.
Which is a terrifying but fascinating place to start. Welcome, everyone, to part 1 of a special, highly detailed 2-part deep dive. If you're listening to this, you already know we're unpacking the heavyweight champion of neuroscience texts, which is Chapter 43 from Principles of Neural Science, the 6th edition.
The definitive guide.
Truly. And our mission today is to thoroughly map the biology of emotion, motivation, and homeostasis. We're going to treat this deep dive as basically a virtual advanced neuroscience class.
I love that approach.
Because before we can understand the pathologies of the brain-like addiction, which is going to be the entire focus of part 2, we first really have to understand the magnificent, complex machinery of normal motivation. So the core question driving us today is, what actually is a motivational state, and how does it orchestrate your goal-directed behavior?
And that rat on the electrified grid forces us to confront a pretty profound reality about how the brain assigns value to the world. We tend to think of motivation as this psychological abstract, like a feeling or just a sense of willpower. But in strict neurobiological terms, it is a highly conserved, rigorous computation.
It's math, basically.
Exactly. And to understand the hack that researchers James Olds and Peter Milner performed with that electrical lever back in 1954, we have to look at the baseline state first. We have to establish the actual biological rules of desire.
I like to think of the brain's motivational circuitry as this highly advanced operating system. It's constantly reweighting the value of every single input based on the background applications currently running in your body and mind.
That's a perfect analogy.
So to establish those rules, we need to look at how the brain filters reality. The textbook opens with this scenario that perfectly illustrates this, which they call the cheetah's dilemma.
Oh, this is a classic framing.
It's so good. So imagine a cheetah resting in the midday sun, just taking refuge in the shade. Out in the distance, an antelope wanders into view. The cheetah looks at it, but registers total indifference.
Zero reaction.
Right? It doesn't orient, it doesn't flinch, it certainly doesn't prepare to move.
But then, you fast forward a few hours to the late afternoon. The exact same cheetah spots the exact same antelope. But now, that visual stimulus provokes an immediate, intense, orienting response.
The whole body changes.
Yeah, the cheetah drops into a low stalk. Now let's analyze the neurobiology of that shift. Because the external stimulus, the antelope, is entirely constant.
It hasn't changed at all.
Exactly. The physical photons hitting the cheetah's retina are absolutely identical. The signal transduction traveling through the optic nerve to the lateral geniculate nucleus and then that subsequent mapping in the primary visual cortex, they are processing the exact same visual data. The physics of the world haven't changed.
The prey hasn't changed. So the valuation of that prey must have changed. And I think this fundamentally defines what a motivational state actually is. It's not just a mood. A motivational state is an internal biological imperative that actively alters the brain's attentiveness, its goal selection, the investment of effort it's willing to expend, and its responsiveness to specific stimuli.
It's an active filter.
It really is the filter through which all incoming sensory reality is judged, dictating whether an animal is going to initiate approach or avoidance.
Precisely. Because the visual cortex might see the antelope, but it's the motivational state that tells the motor cortex what to actually do about it.
Right.
And this state is governed by two interacting variables. You have internal variables and external variables.
Let's break down the internal ones first.
Sure. So for internal variables, we are primarily looking at physiological signals. In the case of the cheetah, the brain is asking, how depleted are its glycogen stores? What is its blood osmolarity indicating about its current hydration levels?
Like, is it thirsty?
Exactly. And what is the status of its circadian clock? Because we know that circadian rhythms heavily dictate foraging behaviors.
Well, that makes sense. Time of day matters.
Absolutely. And you also have to factor in reproductive status. If a female animal is lactating, for instance, her metabolic demands are exponentially higher. That drastically shifts her internal motivational state to seek calories with, frankly, far greater aggression and risk tolerance than a non-lactating female.
But what I find really fascinating here, especially as we start scaling this up to human behavior, is that internal variables are not strictly physiological.
No, they aren't.
They aren't just hunger or thirst or thermoregulation. The text is very clear that internal variables can be deeply cognitive. A cognitive rule can act as the exact same kind of internal driver as a sudden drop in blood sugar.
And the book uses the rules of blackjack to demonstrate this, which is a brilliant framing.
I love that example. It's so relatable.
It's a perfect translation of the cheetah's dilemma straight to the human prefrontal cortex.
So imagine you're sitting at a casino table. The dealer flips over a king of spades. Now, much like the antelope on the savannah, the visual processing of that card is identical every single time you look at it.
Right, it's just cardboard and black ink.
Just a specific shape on your retina. But if your current hand totals 20, seeing that king of spades is going to elicit an immediate visceral feeling of dread, you've busted.
Game over.
The sensory input is evaluated as highly aversive. However, if you're sitting there with an 11 and that exact same king of spades hits the felt, your brain floods with an immediate rush of joy. You hit 21, you win.
And if you track the causality there, the differential valuation of that exact same visual stimulus is entirely dependent on an abstract cognitive construct.
A completely made-up rule.
Exactly. The mathematical rules of blackjack, which are held in your working memory, they act as the internal variable. Your prefrontal cortex is taking a learned, abstract rule and utilizing it to modulate the motivational and emotional centers of your brain, just as surely as the lateral hypothalamus uses a lack of glucose to drive hunger.
The book also provides a highly relatable social example along these lines. Think about the act of chugging a cheap beer or a glass of wine.
Oh, right.
If you do that at a wild college frat party, the social context, which is the internal cognitive variable representing the rules of that specific environment, dictates that the behavior is a fun, rewarding triumph.
You're the life of the party.
Right. But if you take that exact same physical action, drinking the exact same liquid in the middle of a very quiet, formal, Michelin-starred dinner, your brain will process it as a mortifying social disaster.
Which just beautifully demonstrates that abstract human cognition and primitive physiological drive ultimately converge into singular motivational states within the brain.
They use the same hardware.
They do. The complexity of the inputs differs vastly, obviously, between a human navigating social etiquette and a cheetah navigating the savannah, but the final common pathway, that machinery that weighs those inputs and executes the goal-directed behavior, is remarkably conserved across species.
Okay, so we have the internal variables covered, the hunger, the cognitive rules. Now we need to look at the external variables.
The incentives in the environment.
Right. These are the rewarding incentive stimuli out in the world. So let's take our cheetah again. It's late afternoon, the internal variable of hunger is incredibly high, and it is actively stalking the antelope. But as it creeps through the dry brush, this highly dehydrated cheetah suddenly spots a pristine watering hole.
This is where the biological math gets highly dynamic, because the sight of that water is a powerful external incentive stimulus. Suddenly, the brain's circuitry is forced to run a rapid competing cost-benefit analysis.
It has to make a choice.
Yes, it has to weigh the severity of the hunger against the severity of the thirst, while also factoring in the probability of success.
Which is huge.
It is. The antelope hunt costs immense energy and has a really high failure rate. The water, on the other hand, is stationary and guaranteed. So the external stimulus of the water tips the balance, shifting the overall internal state. The cheetah abandons the hunt and interrupts its quest for food just to hydrate.
But the value of that water isn't infinite. And I think this introduces a critical biological modulator here, which is satiation. Because once that cheetah drinks its fill, that external stimulus, the water, entirely loses its motivational power. It ceases to be a reward.
Satiation is the feedback loop that basically prevents the system from spiraling out of control. It modulates the internal variable. Let's look at it in humans with a high-calorie reward.
Like dessert.
Yeah, think of an incredibly rich chocolate cake. That first bite is innately rewarding. It elicits a significant neurochemical response because your system values the sudden influx of dense calories.
Well, it feels great.
Right. But if you are forced to eat an entire cake, your internal state shifts drastically. Your gastric stretch receptors start firing, your blood glucose spikes, and all these homeostatic feedback mechanisms kick in.
I feel sick.
Exactly. So if offered another slice, that innately rewarding external stimulus suddenly becomes highly aversive. You don't want it. Satiation has decreased the reward value to 0 or even pushed it into negative territory.
This constant reweighting of internal deficits against external stimuli is exactly how we survive in the immediate term. It manages what the text calls our short timescale regulatory needs.
The day-to-day survival tasks.
Right. These are the acute, immediate behaviors designed to fix a sudden physiological imbalance. Feeding to fix a caloric deficit, drinking to fix blood osmolarity, shivering or sweating for thermal regulation. You take the action, and the homeostatic balance is restored relatively quickly.
But mammalian brains, and particularly human brains, are not strictly reactive to just immediate homeostatic deficits.
Thankfully.
Yeah, we also have non-regulatory needs, which operate on vastly longer timescales. These are behaviors that serve biological and evolutionary imperatives going far beyond immediate survival. We're talking about finding and sustaining a romantic partner, raising offspring, or, achieving a massive professional goal.
Like surviving a 5-year PhD program.
Exactly. Obtaining a graduate degree is a prime non-regulatory goal. It requires sustained, highly focused behavior over years. And the real biological challenge here is that you have to string together a massive sequence of complex behaviors, and critically, almost none of those individual actions provide an immediate reward.
I mean, not even the cheetah gets an immediate reward. It doesn't just look at the antelope and instantly ingest calories.
There's a whole sequence.
The textbook maps it out nicely. The cheetah must stalk, then it has to initiate the chase, run the animal down, execute the kill, and finally drag this heavy carcass to a refuge to avoid scavengers before it can even take its first bite.
That is an enormous expenditure of ATP.
And a massive risk of injury.
All happening before a single calorie is restored. So how does a biological system sustain that kind of delayed gratification?
Right.
It sustains it through the neurological mechanism of what we call incentive stimuli.
Let's unpack that.
These are environmental or internal cues that bridge the temporal gap between the exertion of effort and the final distant reward. They don't provide the caloric or homeostatic payoff themselves, but they signal to the nervous system that the long-term goal is achievable.
Like breadcrumbs.
Kind of, yeah. When the cheetah successfully stalks 10 yards closer without snapping a twig and alerting the herd, that positional progress acts as an incentive stimulus. It provides a micro-reward that sustains the motivation to initiate the next phase of the sequence, which is the sprint.
I always conceptualize this for long-term human goals by comparing it to massive, notoriously difficult video games.
Oh, I like this.
If you ever played a game that requires you to navigate a brutally hard level where you just die repeatedly, the only reason you don't throw the controller against a wall and quit is because of the save points or the progress bars.
That is a highly accurate structural analogy.
Because when you reach a save point in a game, you haven't won. You don't get the final reward, you don't beat the boss, you don't finish the story. But that small visual or auditory chime on the screen is biologically essential.
Your brain loves it.
It does. Your brain registers it as an incentive stimulus. It's a localized neurochemical hit that says, hey, your effort was not wasted. You are making measurable progress toward the distant goal. Maintain your investment of energy.
Because without those safe points, without those progressive signals, the temporal gap between the effort and the reward becomes too vast, and the entire motivational system collapses.
You just drop the controller, or you quit the PhD.
Exactly. And if we understand that the brain absolutely requires these progressive signals to string behaviors together across time, it forces us to look under the hood. Where exactly in the neuroanatomy is this evaluation taking place?
How does the brain physically compute that a specific stimulus is worth all that effort?
Which brings us right back to that rat on the electrified grid.
Right, the Olds and Milner experiment.
Because to understand the physical architecture of desire, we need to rigorously define what a reward actually is in neurobiology. Because it's not just a subjective feeling of liking something.
No, in the neurobiological framework, a reward has a very strict operational definition. A reward is an object, a stimulus, or an activity that possesses positive value, and crucially, it incites an animal to switch from one behavior to another, or to resist the interruption of an ongoing action.
The text provides a great ethological example here. Imagine a rat foraging in its natural environment. It's exploring, mapping the terrain, sniffing for predators. Suddenly, it uncovers a high-calorie seed.
A jackpot.
Right. The seed has positive biological value, so this reward incites the rat to immediately switch behaviors. It ceases its exploration and initiates feeding. Furthermore, if a competing rat approaches and tries to steal that seed, our rat will actively defend it.
He'll fight for it.
Exactly. The reward incites the animal to resist interruption of the ongoing feeding behavior.
And making that decision to stop exploring, to eat, and to fight another rat requires a really rigorous cost-benefit analysis by the neural circuitry. The brain has to continuously link the sensory representation of a potential reward to its current physiological needs, while simultaneously calculating environmental risks.
Like the antelope straying from the herd, the benefit there is access to denser, ungrazed grass. The cost is the loss of the herd's collective vigilance and a massively increased risk of cheetah predation.
And, conversely, the cheetah deciding to launch an attack on that stray antelope calculates the benefit of a massive caloric intake against the severe metabolic cost of a sprint, plus the potential for debilitating injury if the prey fights back or escapes.
And the neural mechanisms responsible for executing this high-stakes cost-benefit analysis were just blown wide open by James Olds and Peter Milner in 1954. This brings us to intracranial self-stimulation, or ICSS.
This is arguably one of the most consequential and frankly visceral experiments in the history of behavioral neuroscience.
It completely shifted our understanding of the brain's motivational centers. What Olds and Milner did was implant microelectrodes directly into specific targeted regions of a rat's brain.
Directly into the tissue.
Right. They then placed the rat in an operant conditioning chamber, basically a box with a lever. But pressing this lever did not dispense a pellet of food or a drop of water.
Like a normal experiment would.
Exactly. Instead, it triggered a brief, localized electrical pulse directly through the implanted electrode into the brain tissue.
And the behavioral results were entirely unprecedented. The avidity, the sheer relentless persistence of this self-stimulation defied all natural ethological logic.
The drive to obtain this electrical stimulation overrode fundamental survival imperatives. As we discussed at the very start of the show, these animals would cross highly aversive electrified grids, absorbing painful shocks to their feet just to reach the lever.
They'd run uphill over physical hurdles.
They would press the lever at astonishing rates, sometimes thousands of times an hour for hours and days on end, entirely foregoing biological needs like water and food. Even a starving rat, given only a brief temporal window of access to food each day, would ignore the food completely to press the lever for brain stimulation.
They would literally stimulate themselves to the point of exhaustion and starvation. The phenomenon is known as brain stimulation reward, and it elicits a motivational state so overwhelmingly powerful that it obliterates the cost-benefit analysis of survival. So, anatomically speaking, where were these electrodes actually placed? What specific wiring were they hijacking?
Through systematic mapping, researchers found that while the circuitry is broadly distributed, stretching from the olfactory bulb all the way down to the brainstem, the most sensitive and effective sites, the ones that drove this heedless compulsive pursuit, were found along a pathway called the medial forebrain bundle, and specifically within the ventral tegmental area, or VTA, located in the midbrain.
The VTA is the absolute star player in this circuitry. When we talk about motivation and reward, We're almost always tracing pathways back to or from the VTA.
Yes, because the VTA is the origin point for a massive network of dopaminergic neurons. When Olds and Milner stimulated the VTA, they were causing massive artificial action potentials in these dopamine-producing cells.
Forcing them to fire.
Exactly. These neurons project their axons outward to several critical target regions. They send dense dopaminergic projections to the nucleus accumbens, which is the major component of the ventral striatum, and a critical hub for processing reward and assigning incentive value.
They also project to the ventromedial head of the caudate nucleus in the dorsal striatum, right?
Right, which is highly involved in habit formation and motor action. And importantly, they send pathways to the basal forebrain and ascending projections directly into the prefrontal cortex, heavily influencing executive function and decision-making.
So the VTA is essentially a central broadcasting station. It sits right in the midbrain, evaluates incoming signals, and then broadcasts dopamine upward into the striatum and the cortex, basically saying, hey, this is important, pay attention, move toward this.
That's a great way to summarize it.
But what tells the VTA to fire in a normal, unhacked brain? It doesn't just spontaneously release dopamine for no reason.
No, it is highly regulated by complex afferent inputs. These dopaminergic neurons are excited by glutamatergic cells coming down from the prefrontal cortex and the amygdala.
Okay, so top-down signals.
Exactly. This means your high-level executive center, which is evaluating goals and rules, and your emotional processing center are actively sending excitatory glutamate signals, telling the VTA to fire. Simultaneously, they are intensely excited by cholinergic cells neurons releasing acetylcholine that project upward from the dorsal, tegmental, and pedunculopontine nuclei in the hindbrain.
And like any finely tuned engine, there has to be a braking system, right?
Always. They are kept in check or inhibited by local GABAergic interneurons that prevent runaway firing.
I really want to highlight that cholinergic input from the hindbrain, because the text makes a crucial point here that I feel often gets lost. In pop science, dopamine is treated as this solitary magical molecule.
Like it does everything alone.
Yeah, but the textbook emphasizes that if you pharmacologically block that cholinergic input from the hindbrain, you actively reduce the rewarding effects of the ICSS electrical stimulation. The rat simply won't press the lever as hard.
That is a vital mechanistic detail.
Yeah.
It proves that this circuitry is deeply physically interconnected. It is not a monolithic dopamine-only pathway.
It's a network.
It relies on a delicate, synchronized symphony. You have top-down glutamate from the cortex, bottom of acetylcholine from the hindbrain acting as an amplifier, local GABA acting as a brake, and then dopamine acting as the final broadcaster. Altering any one of those neurotransmitter systems disrupts the entire motivational computation.
Okay, I had to step back and look at the evolutionary logic of this because it feels like a total paradox to me.
Oh.
Well, we have this incredibly sophisticated, finely tuned, cost-benefit circuit involving all these different brain regions and neurotransmitters, designed over millions of years of evolution to keep us alive. But a starving rat will press a lever for a tiny electrical shock until it literally dies of starvation. Hasn't the system completely failed?
It looks like it.
To use an analogy, if you hack a thermostat to keep the furnace running until the house burns down, the thermostat failed its one job.
I like the thermostat analogy, but we need to adjust how we view the hack itself. It's not that the thermostat failed to read the temperature, it's that Olds and Milner didn't just trick the sensor.
What do they do then?
They took a pair of wire cutters, snipped the sensory cables entirely, and spliced a battery directly into the control board, feeding it a constant electrical voltage that screamed, "The temperature is perfect, keep the furnace on."
I see.
In nature, a reward is intrinsically coupled to physiological homeostasis. The animal eats the seed, the complex carbohydrates are broken down, blood glucose rises, insulin is released, leptin signals from fat cells increase, and a true biological deficit is met. All of those peripheral signals feed back into the brain to naturally modulate the VTA.
A natural feedback loop.
Exactly. What ICSS does is completely uncouple the signal of reward from the biological reality of homeostasis. It bypasses the sensory inputs, bypasses the digestive feedback loops, and directly, artificially forces the exact neurons that signal this is the most important thing for survival to fire.
So the biological system didn't actually fail.
No, it performed exactly as it is anatomically designed to perform when given that specific electrical command at that specific synapse. The experiment just reveals the raw, unmitigated power of this circuit when it is forcefully decoupled from natural homeostatic limits.
And this brings us right to the threshold of part 2 of our deep dive, because addictive drugs do chemically exactly what those electrodes did electrically.
It's the same mechanism.
The text points out that if you give a rat cocaine or nicotine, it actually lowers the threshold for this brain stimulation reward, meaning an animal given cocaine will enthusiastically press a lever for a tiny microelectrical current that it would normally just ignore as too weak.
The drug physically amplifies the sensitivity of the reward circuit.
Which naturally leads us to confront one of the most pervasive, culturally entrenched misconceptions in all of popular neuroscience.
Oh, this is a big one.
The hedonic myth. The persistent, completely flawed idea that dopamine equals pleasure.
It is a view of dopamine that dominated early neuroscience and unfortunately still heavily dominates the public consciousness today.
It's everywhere.
The original hypothesis was that dopamine conveyed a hedonic signal in the brain, that the release of dopamine was the direct physiological cause of subjective pleasure or joy.
I mean, if you open any social media app right now, you will see this everywhere. You'll see dopamine fasting, dopamine menus. People use the word dopamine as a direct synonym for happiness or having a good time. They think eating a donut releases dopamine, which makes you feel good, and therefore you want another donut.
But neurobiologically, the empirical evidence has dismantled this theory entirely. The mechanisms of dopamine are far more complex and mathematically precise.
Let's break that down.
First, we now have extensive data showing that dopamine neurons can be heavily activated by aversive stimuli, as well as rewarding ones.
Oh, really aversive.
Yes. If a rat is exposed to a stressor or a painful shock, certain dopamine pathways light up. If dopamine were purely a pleasure molecule, that physiological response would be impossible.
But I think the truly definitive proof comes from the depletion studies. The textbook details experiments using knockout mice and neurotoxins that are just mind-blowing if you still believe dopamine is pleasure. Let's walk through the 6-hydroxydopamine lesion studies.
Let's do it. 6-hydroxydopamine, or 6-OHDA, is a highly specific neurotoxin. When it's injected into the brain, it is taken up by the dopamine transporters and it selectively destroys dopaminergic terminals.
So it wipes out the dopamine.
Effectively stripping the animal of its dopamine pathways while leaving other systems intact, yes. Scientists also used genetically engineered knockout mice that completely lack the gene required to synthesize dopamine in the first place.
Right.
Under the hedonic theory, if dopamine equals pleasure, these dopamine-depleted animals should be entirely incapable of experiencing pleasure. They should exhibit complete anhedonia.
But they don't. The textbook explicitly states that these dopamine-depleted animals continue to exhibit normal, hedonic, pleasurable physical responses to sweet substances like sucrose.
That's the critical finding. When researchers place a drop of sucrose on the tongue of a dopamine-depleted rat, the animal still displays the exact same ethological facial expressions of liking the sugar as a normal rat.
What do those expressions look like?
They show rhythmic tongue protrusions, lateral tongue movements, relaxed facial muscles. They clearly still enjoy the taste. The subjective experience of pleasure remains completely intact without dopamine.
So if dopamine isn't producing the pleasure, what is?
Pleasure, the subjective state of liking something, is not mediated by a single neurotransmitter in a single pathway. It is processed by a broadly distributed network of what we call hedonic hotspots.
Hedonic hotspots.
Right. This network spans sensory cortices, processing the taste and smell, the association cortex, the prefrontal cortex, specifically the orbitofrontal regions which heavily evaluate reward valence, the amygdala, the hippocampus, the Nucleus Accumbens, and the ventral pallidum.
That is a massive network.
It is. And neurochemically, this liking network relies heavily on opioid and endocannabinoid signaling, not dopamine.
So we have a physical neuroanatomical separation between liking a reward and wanting a reward. This feels like a massive paradigm shift.
It really is the foundation of modern reward theory, pioneered by researchers like Kent Berridge. Liking is the hedonic pleasure mediated by those hotspot networks and opioid and cannabinoid signaling.
And wanting.
Wanting, on the other hand, is what we term incentive salience. It is the intense biological drive, the craving, the motivation to pursue the object. Dopamine drives the wanting. Dopamine acts as a teaching signal, stamping in the value of the cues that predict the reward and motivating the animal to seek it out again.
You know, this distinction is really the only way to explain the realities of human behavior, especially in the context of severe pathology.
Absolutely.
If dopamine was just pleasure, then someone struggling with severe addiction would simply stop taking a drug when the drug stops being pleasurable. But we know that is absolutely not what happens in the real world.
Exactly. The hedonic principle completely fails to explain the persistence of drug use as negative consequences mount and tolerance builds.
People persist in behaviors that destroy their health, their families, their entire lives. They often report that they don't even like the drug anymore.
Right, the high is gone.
The high is gone, replaced only by relief from withdrawal or just this overwhelming compulsion. The pleasure network, the liking, has downregulated. But the mesolimbic dopamine system is still firing massively, screaming at the prefrontal cortex that they still want it, that it is essential for survival.
The incentive salience is pathologically amplified while the hedonic pleasure is blunted. So if dopamine isn't pleasure, but rather a driver of wanting and learning, we have to look precisely at what information those VTA dopamine neurons are actually transmitting. What is the mathematical language of this learning signal?
This brings us to one of the most elegant discoveries in modern neuroscience, the Schultz experiments,
Brilliant work.
which really revealed the mathematics of prediction error. Wolfram Schultz and his colleagues completely changed how we understand dopamine by looking at the exact millisecond timing of these neurons firing.
Let's walk through this carefully, phase by phase, because this isn't just biology. It is a rigorous, subconscious biological algorithm happening in your midbrain right now. Schultz was recording the action potentials, the firing rates of individual midbrain dopaminergic neurons in awake active monkeys.
Okay, so phase 1 is the unpredicted phase. The monkey is just sitting in the chair, no cues, no tasks. Suddenly, without any warning, a small tube delivers a drop of sweet fruit juice directly into the monkey's mouth.
We have to look at the baseline first. These dopamine neurons typically fire at a slow, steady baseline rate, maybe 3 to 5 hertz. In phase 1, when that unpredicted drop of juice arrives, the unexpected reward elicits a sudden transient burst of firing, spiking up to 20 or 30 hertz.
So it jumps up.
Yes. The brain registers a massive positive signal. Something unexpectedly good and biologically valuable just happened.
Then we move to phase 2, which is the predicted phase. This is where classical conditioning comes in. The monkey is trained that a specific environmental cue, let's say a flash of light on a screen, or a specific auditory tone, will be followed exactly 2 seconds later by a drop of juice. And the monkey learns this association perfectly.
This is where the true function of dopamine reveals itself. As the monkey learns that the light predicts the juice, the timing of the dopamine burst physically shifts.
It moves.
It does. The neurons no longer fire a burst when the actual juice hits the tongue. Instead, the burst of firing moves backward in time, occurring at the exact millisecond the predictive cue, the light or the tone appears.
The dopamine spike shifts from the reward to the cue that predicts the reward.
Exactly.
And here's the mind-blowing part to me. When the juice actually arrives 2 seconds later, right on time, the dopamine neurons maintain their slow, steady baseline firing. There is zero spike when the sugar is actually consumed.
Because the reward occurred exactly according to the brain's internal prediction model, the environment matches the expectation perfectly. There is no error.
Which brings us to the critical phase 3, the disappointment phase. The monkey is fully trained. The cue is presented. The light flashes. The monkey gets the expected dopamine burst at the cue, signaling reward is imminent. But this time, the researchers trick the monkey. They withhold the juice.
And if you look at the electrical recording of the dopamine neuron, right at the exact millisecond the juice was mathematically expected to arrive based on the timing of the cue, the baseline firing actually pauses.
It just stops.
The neuron goes silent. The firing rate drops significantly below the baseline for a fraction of a second.
This is the biological manifestation of disappointment. It's called the prediction error signal. It is a subconscious, continuous mathematical equation. A dopamine burst above baseline means better than expected, a positive prediction error. A pause, a drop below baseline, means worse than expected, a negative prediction error.
And steady baseline firing means the environment is exactly as predicted, no better, no worse.
I want to make this intuitive for you listening. Let's map this biological algorithm onto a human scenario. Let's use an analogy about your job and your year-end bonus.
Okay, let's hear it.
So phase 1, you've just started at a new company. You have absolutely no idea bonuses are even a thing here. Suddenly, on a random Tuesday, a $5,000 bonus hits your bank account. Total surprise.
Best Tuesday ever?
Right. Your brain experiences a massive positive prediction error, better than expected. You get a huge burst of dopamine, reinforcing that this job is highly valuable.
That's the unexpected juice.
Exactly. Now, phase 2. Fast forward a year, you now have a contract that explicitly guarantees you a $5,000 bonus on December 1st. Now, the dopamine spike happens when you get the confirmation email from HR on November 15th. That email is the predictive cue.
The flash of light.
Yep. When December 1st rolls around and the money sits in your account, you're pleased, the hedonic liking network is happy you have money, but your mesolimbic dopamine system stays at baseline. It's exactly what was predicted. No new learning required.
And phase 3.
Phase 3. The following year, you get the HR email, which is the cue, and you get the dopamine spike predicting the reward. But on December 1st, the expected check bounces. The money isn't there.
Oh, wow. At the exact moment you realize the money is missing, your dopamine neurons pause their firing. You experience a visceral, immediate drop in your physiological state.
A total gut punch. That is the negative prediction error, that momentary silence in dopamine firing when the expectation is violated.
I want to emphasize the profound nature of that pause. This is not a human feeling of sadness. It is a rigorous biological algorithm designed to modify future behavior to maximize resources. The brain uses these prediction errors to update its internal models.
So it's a learning mechanism.
It is. If a reward is exactly as expected, Schultz's model posits that as long as nothing changes in the environment, there is nothing new to learn. Therefore, there is no need to alter behavioral responses and no need to expend the energy of a dopamine spike at the time of the reward. But that pause, that negative prediction error, is the brain saying, the model is wrong. The cue lied. Devalue this pursuit in the future.
Do not trust that cue next time.
Yep.
The environment has changed. Update the behavioral model.
Exactly. The alterations in dopamine release, the bursts and the pauses, are not about pleasure. They're a continuous calculus designed to modify future responses, to maximize the likelihood of obtaining vital resources, and to minimize fruitless, energy-wasting pursuits.
It is an incredibly elegant, mathematically beautiful system. But as we move into the final stretches of this first part of our deep dive, we have to acknowledge that biology is rarely a perfect monolithic equation.
That's very true.
As neuroscience advances, we are seeing immense complexities that set the stage for how this system could become pathological.
Yes. While the prediction error model is the foundational bedrock of how we understand reward learning, recent data shows the system is vastly more heterogeneous. For instance, we discussed the dopamine-deficient knockout mice earlier. While they still like rewards, the fascinating addition is that under certain specific conditions, these mice without dopamine can still learn where to find a sugar or cocaine reward in a maze.
Without dopamine.
Yes. This suggests that while dopamine is the primary driver of incentive learning, it might not be strictly 100% required for all forms of spatial reward learning.
And we also see pharmacological evidence that separates learning from wanting. If you look at rodents given systemic amphetamines, which forcefully elevate presynaptic dopamine levels over extended periods, they exhibit a massively enhanced wanting behavior.
They crave it more.
Right. They will respond much more intensely and frantically to Pavlovian cues that predict a sucrose reward. The drug amps up the incentive salience, the drive, without necessarily altering the underlying learning of the cue itself.
What researchers are mapping right now is the extreme heterogeneity of midbrain dopamine neurons. We used to think of the VTA as a monolithic block, a cluster of identical neurons all doing the exact same prediction error math in unison.
But they aren't.
No. Using advanced recording techniques, we now see distinct subpopulations. Some dopamine neurons are activated by both rewarding and aversive stimuli. They seem to signal general salience or alertness. Some prefer one over the other, and some have strictly opposite responses. They are activated by rewards and entirely suppressed by aversive stimuli, fitting the classic Schultz model perfectly.
Okay, this raises a massive logistical problem for the brain. If you have dopamine neurons that are this heterogeneous, responding to totally different, sometimes contradictory stimuli, some firing for sugar, some firing for a painful shock, and they are all releasing the exact same neurotransmitter, dopamine, how does the broader brain not get hopelessly confused?
It's a great question.
How does it know if a dopamine pulse means run toward this or run away from this?
That is the cutting edge of the field, and it comes down to precise topographical mapping and circuit specificity. It's all about the physical wiring. There is evidence that these functional differences in dopamine neurons are intrinsically related to differences in their afferent inputs, where they are receiving signals from, and their efferent projections, where they are sending signals to.
The wiring diagram dictates the meaning.
Precisely. A dopamine neuron in the VTA that fires in response to an aversive shock might receive its input primarily from a specific stress-related nucleus in the brainstem, and it might project its dopamine exclusively to a very specific subregion of the prefrontal cortex or the amygdala dedicated to threat avoidance.
Okay.
Meanwhile, a neighboring dopamine neuron that fires for a sugar reward receives input from the lateral hypothalamus and projects down into the shell of the nucleus accumbens to drive approach behavior. The brain keeps the signals distinct through dedicated parallel wiring channels, even though the chemical messenger dopamine is absolutely identical.
The microarchitecture is just staggering. But this complexity, this beautifully balanced parallel wiring system, brings us to the crucial contrast between natural rewards and drugs of abuse. This is the pivot point for everything we will discuss in our next session.
Yes. For natural rewards like the sweet juice for the monkeys or a calorie-dense meal or social interaction, once the environmental cues are learned, the dopamine firing returns toward baseline levels upon consumption. The learning algorithm stabilizes.
The biological deficit is met, the system updates, and homeostasis is maintained.
Exactly. But addictive drugs do not play by the rules of homeostasis. They completely bypass that natural algorithm.
Addictive drugs are basically chemical Trojan horses.
That's exactly what they are. Unlike a natural reward, which relies on sensory feedback to modulate the VTA, an addictive drug artificially, pharmacologically forces dopamine release in the reward circuitry no matter how often it is consumed.
Every single time.
The magnitude of this forced release is often vastly greater than anything seen with natural rewards. And crucially, because it is a pharmacological action right at the synapse, this dopamine is released even when the drug is entirely expected, and even when it no longer produces subjective pleasure.
Let's tie that back to the math. What are the profound implications of this chemical reality on the Schultz prediction error equation?
It creates a mathematical loop of infinite desire. To the brain's learning algorithm, the consumption of an addictive drug always, biologically, registers as a massive positive prediction error.
It breaks the math.
It does. Even if the person knows exactly what the drug will do, the chemical forces the dopamine spike. It always signals better than expected. Therefore, it endlessly, relentlessly updates the learning algorithm, stamping in the value of the drug higher and higher, forcing the brain to prioritize drug seeking over all other survival pursuits, because the biological math is telling the cortex that this substance is the most valuable thing in the universe.
The algorithm is broken by a permanent positive error. We need to take a breath here because we have covered immense ground today.
We really have.
Let's synthesize what we've unpacked in this first part of our deep dive. We've explored the incredible, finely tuned biological machinery that orchestrates every goal-directed behavior you undertake. We've seen how a motivational state is not just a mood, but an active physiological filter, seamlessly integrating your internal deficits like thirst or a cognitive rule in a casino with external environmental cues.
And we've mapped out the physical circuitry, from the inputs to the VTA, broadcasting dopamine outward to the nucleus accumbens and the prefrontal cortex. We've debunked the enduring myth that dopamine is merely a pleasure chemical, revealing it instead as a rigorous, mathematically precise prediction error signal.
It's a teaching signal.
It is a teaching signal that drives our survival and long-term ambitions by bridging the temporal gap between immense effort and distant rewards.
It is a system exquisitely designed by evolution to keep you alive and moving forward. But as we prepare for part 2, I want to leave you with a final provocative thought, a philosophical and biological cliffhanger based on everything we've just learned.
I'm ready. Go ahead.
If our entire framework for goal-directed behavior, our very sense of motivation, our ability to prioritize taking care of our children or going to work or simply surviving, if all of that relies on a delicate prediction error equation that is supposed to return to baseline once a lesson is learned, what happens to human free will when a synthetic molecule artificially forces that equation to permanently, endlessly equal better than expected?
That is the exact question that defines the absolute tragedy of the addicted brain.
When you look closely, you see a motivational system that hasn't just broken down, but has been ruthlessly chemically reprogrammed to desire its own destruction. That is exactly what we will be covering next time. This has been part 1. Join us for the next deep dive, where we will explore the lasting molecular, cellular, and circuit adaptations that define the pathological state of drug addiction. See you then.